please join me in welcoming Professor Robin May.
Fantastic. Thank you so much. Thank you for coming. Those of you here in the audience, thank you for joining online. Those of you at home, and in particular, those of you at home, if you find this terrifically boring and turn off, I'll never know. Those of you in the hall, if you find it terrifically boring, you either have to snore quietly or sneak out the back, and I will know. So for those of you who are coming back to this series, we've been talking about immunity, and today we're going to talk about vaccines. For those of you who haven't been before and have absolutely no idea about anything in the rest of the series, it doesn't matter, because this should hopefully stand on its own. And the question I want to address today is about vaccines, how they work and what they might do in the future. And by, I'd like to say by orchestrated design, but actually by complete happy coincidence, today is actually Vaccine Engagement Day. So if you want to learn more about vaccines than you ever thought you would want to know, you can visit this wonderful page from the British Society for Immunology after the lecture, or even during the lecture if it's boring, and find out all sorts of stuff about that. And if you're a social media guru, you can use the hashtag and find out what people are saying about vaccines. What a good bit of timing. So today, for the lecture, what I want to do is think a bit about how vaccines work and maybe how we can do things differently with them in the future. But before we think about how vaccines work, I'm sometimes asked this question, which is not how do vaccines work, but do vaccines work? And that is a very short lecture, because the overwhelming answer is yes, they most definitely do. And let me demonstrate just how much they do. So, for example, the WHO estimates that since the kind of wide rollout of global vaccines, they've saved about 154 million lives. And this is almost exactly my lifetime at the moment. So during my lifetime, vaccines have saved basically twice the entire population of the United Kingdom, which I think is quite remarkable. And actually, when you think about that in the scale of other medical advances like organ transplants or even things like antibiotics, actually, this is one of the most fundamental changes in public health ever. So they absolutely work. And if we look kind of more parochially just at the UK, just a couple of examples here. So this is vaccines for diphtheria and for measles. And you can see this absolute plummet in the rate of those diseases when vaccines are first introduced. And you can do this for, literally tens of diseases. And actually, some of you in the audience who may be of a certain vintage will probably recall being at school and knowing, for example, people who had disabilities or paralysis from polio. You might recall people who had severe scarring from smallpox. And those, of course, were the lucky ones. There were many people who didn't make it through that. Things which actually, if you're on the other side of that life expectancy limit, I'm looking at all the young people in the audience, you have no idea what I'm talking about. And that is great, right? Because those diseases are things that we just don't think about, most of us, most of the time, because of vaccines. So vaccines have been enormously instrumental. They've changed the course of history. And so it wouldn't be kind of right to talk about vaccines without spending a couple of minutes talking about how they came about in the first place. And actually, the history of vaccines is quite an interesting one, because unlike a lot of things in science, there wasn't this kind of sudden eureka moment when we didn't know about stuff and then we didn't. There wasn't the kind of Albert Einstein, God, guess what? We've got all this wrong previously. There was a kind of incremental increase in knowledge generally, with a couple of really landmark events. And the big landmark event that, at least here in sort of Western Europe, we always think of as the kind of the beginning of vaccination is this chap here. So this is Edward Jenner, who was a doctor down in Gloucestershire. And in the late 1700s, he became aware of the observation that others had made, actually, that cowgirls, girls primarily at that time, who milked cattle, would often get this quite mild disease called cowpox. And you can see in this painting, here is the aforementioned cowgirl with her cows in the back, just to remind you of the story. And they'd often get this mild disease called cowpox. But then if smallpox came, which it did quite frequently in those days, they would generally survive unharmed. And in a bit of a kind of stroke of genius, he thought, well, maybe there's some relationship between here. And in this famous anecdote, which I'm sure you all heard about at school, he inoculated in 1796, 14th of May, in fact, this young chap here, James Phipps, who was a young child in the village, with pustules from cowpox. And I think people often say, oh, he inoculated James Phipps. And you think, oh, that's all marvellous. Probably wasn't so marvellous for James Phipps, actually, because there were no hypodermic needles in those days. So actually what he's doing here is essentially scratching the skin until it bleeds and rubbing in cowpox. So it probably wasn't as pleasant as, he's either a very stoic young chap here, or this is a bit of kind of airbrushed history in this painting. Anyway, he did that. And then a couple of weeks later, he came back and he deliberately exposed James to smallpox. And James survived. And in fact, because scientists like to do things lots of times, he did it a few more times, and every time James survived, which I think raises a lot of slightly dubious, to say the least, ethical questions. Fortunately for history, he was right. James survived to a ripe old age. And the point that you could inoculate someone and protect them against disease was proven. And actually, it's because of that that we call these things vaccination, because vaccinate is the Latin for cow. So actually, when you go and have your vaccinations, you're being kind of cowified or catalified or something. And so we often think about Jenner as the kind of the progenitor of vaccination. And in fact, there is an institute, the Jenner Institute, that is devoted to vaccine development. But he did not act alone. And in fact, he was building on a lot of prior knowledge. And sometimes I think some people in history are kind of slightly overlooked. And actually, one of the characters that I think is often overlooked in this story is this lady here, looking very elegant. This is Lady Mary Montagu, who is quite a fun character. I recommend on your journey home tonight, you Google her and find out she was quite the adventurer. And as her name suggests, she was a member of the aristocracy. And she went off with her husband, touring what was then the Ottoman Empire, so now sort of Turkey and that part of the world. And whilst she was there, she had observed that local people did this practice called variolation. And variolation was the process when if someone had got smallpox and had not died and was recovering, they were covered in these horrible pustules, but they were getting better. And local people would take those pustules and they would scratch them into the skin of other people in the hope that it would give them a mild case of smallpox. And they knew that if they had a mild case of smallpox, they would recover, and then they would never get the fully virulent lethal smallpox afterwards. And so this process of variolation is a type of vaccination. And so actually, Lady Mary and many other people actually at the time had sort of seen this process in advance. And in fact, she is instrumental in bringing that practice back to what is now the UK. The slight problem, of course, with variolation is that you're inoculating someone with smallpox, and even if you only give them a mild case, that's great, but sometimes you gave them severe smallpox, and quite often they died. So it's not really a recommended route for vaccination, and we'll see a little bit later about how that's influenced kind of modern thinking around vaccines. So that's my potted history of vaccines, and you can learn a lot more about that if you want to online. But tonight, what we're really thinking about is how do vaccines work? And actually, the way that vaccines work is exactly the same, right? So what Jenner did all those years ago is precisely the same to a first approximation as to what is happening today if you go and have your flu vaccination or travel jabs or whatever it is. And so what you're basically doing when you vaccinate someone is you're triggering an immune response. You're mimicking an infection and your body is responding. So the first thing that happens when you stick a needle in somebody's arm and inject them with a vaccine is you trigger white blood cells to come to that site. They're called in the way you see here. This is actually not a vaccination. This is a wound. And those of you who've been to my lectures before might have seen this video. But you can see these little white blood cells zipping out of the blood circulation and coming to this site because they have recognized that something foreign is there, something that shouldn't be in the body has appeared. And that might be an infection, a bacteria or whatever. In the case of a vaccination, it's the thing you've injected, the vaccine you've injected. And this is the reason why vaccines, unfortunately, usually hurt a bit, because you've stabbed the needle into someone and you are driving an inflammatory response, right? So if you have a vaccine, typically, the needle hurts, but also it remains sore for a couple of days. It quite often goes red and hot because you'll have an underlying inflammation reaction. And sometimes you get a bit of a fever because the body is responding to the vaccine as if you've had a disease, but of course you don't have the disease. And that's quite important, actually. You need to trigger that immune response. A vaccine that doesn't do any of those things is probably not going to provoke a strong enough immune response to protect you later on. But what's actually happening on a kind of cellular level? How do these things actually go on to protect you? And this is one of the most kind of beautiful, in my mind, beautiful and kind of elegant ways that cells work to protect us from infection. And the key thing about the sort of response to a vaccine is it relies on two independent signals that converge to a single response. So here's the first one. The first one is you've injected into somebody some bits of a foreign molecule. So let's say it's a bit of a virus, for example. Those bits of a virus are picked up by cells that are close to the site of injection called antigen-presenting cells. And so there are several different types of those. The one that you have here is called a dendritic cell. You can see it wildly flailing its arms around, picking stuff up. In this case, this is not a vaccine. This is actually a big fat yeast particle. But the same principle applies if this was a bit of a viral protein, for example. These cells pick up that protein. And they do that, they chew it up, and then they present it on their surface to the rest of the immune system. They are professional alarm raisers, if you like. They're waving it around and saying, hey, rest of the immune system, I've seen this thing, definitely not human, need to deal with this, okay? And they do that by talking primarily to a type of cell, white blood cell called a T cell, which is a smaller cell down here. So they wave this thing around and they say, okay, T cell, I've seen this, you need to kind of start getting your act together. And you can see that happening here. So in this movie, the green cells are those dendritic cells, and the yellow cells are T cells. And what you see is the dendritic cells doing their waving stuff, like some kind of, dad dancing on the floor or something. And the yellow T cells zip past and occasionally get sort of stuck on the surface for a couple of minutes and talk to each other. What is happening here is that the dendritic cell is trying to find a matching T cell to what it's displaying on its surface. So in your body, you have literally millions and millions of T cells. And one of the remarkable things about those T cells is that as they have developed, each of them has developed a receptor on its surface that will detect a different foreign molecule. Okay, so you start off with T cells and they can recognize all sorts of things. They shuffle and shuffle and shuffle and they come up with a receptor that recognizes a particular molecule, say a little bit of a protein. Each T cell does that separately. So within your millions of T cells in your body, each one recognizes a different sort of protein. And so you have the capacity to recognize literally millions of different proteins. And so back in that movie that I just showed you, what's happening is the dendritic cell is waving this little bit of a virus around, and T cell after T cell is coming past going, nope, I don't recognize that. Nope, I don't recognize that. Nope, nope, no, 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 no. And eventually one comes past and goes, oh yeah, that's the one I recognize. And becomes primed, becomes activated. It's a bit like, I don't know, trying to find a kind of key for your front door and you've got 25 keys, you keep going, and eventually you get one and you go, okay, now I'm in. So the dendritic cell has been waving its virus around. It's talked to hundreds of T cells who are presumably very bored, but the one T cell that says, oh yeah, I recognize that, is now activated. So that's signal number one, right? Signal number two is happening at the same time with another sort of white blood cell, and that's this one, the B cell. Okay, so T cells and B cells, that's it for the alphabet tonight, I think. And B cells, in some senses, are very similar to T cells. So they also, during development, produce a receptor on their surface that recognizes different sorts of molecules. So you have millions of B cells too, each one recognizing a different sort of molecule. And so these, though, don't need an additional cell to help recognize something. They're sitting in your circulation, wandering around your body, actually, as we speak, and they're permanently on the lookout for something that will fit this receptor on their surface. So what's happening, even as you're here listening or possibly being bored by my lecture, is that stuff that you are inhaling or eating or you've got through your cuts or whatever is bumping around in your circulation. And a lot of that stuff does not recognize B cells. It's bouncing off, doesn't fit your receptor. B cell is uninterested, off it goes. But if something in your body does fit that receptor, now the B cell becomes activated. It kind of gets woken up, okay? And again, millions of your B cells will not recognize that particular protein, but the one that does becomes activated. So let's imagine I've given you a vaccine against a particular virus, say polio, for example. Most of your T cells uninterested, but the one that recognizes polio has woken up. Most of your B cells uninterested, but the one that recognizes polio has woken up. And now you have these two signals together, and now it gets really clever. So that B cell that recognized that, what we call an antigen, that foreign protein, that polio protein, eats it, takes it in, chews it up, and then it puts it back out onto its surface. And this tells the rest of the body, okay, I'm primed now, I've woken up, I recognize my antigen, and this is what I've seen. So I've recognized there's trouble here, and I've seen this. Elsewhere in your body, the T cell that was responsive to the same thing has also become activated. He's wandering around, now really looking for that same antigen. And when he meets it, now all hell breaks loose. Because now this guy who recognizes polio and this guy who recognizes polio get together, and the T cell essentially activates the B cell. It says, unleash everything you've got, which is what the B cell does. You might think to yourself, gosh, this is a completely complicated system. Why on earth do we just, why do we need two types of cell? Why do we need all this complexity? And the reason, of course, is that you don't want this to inadvertently fire. And I don't know if anyone's a kind of fan of sort of, Cold War thrillers, for example. So if you love these kind of, like Hunt for Red October or these kind of, nice kind of sort of Cold War-based things, you will know that no one person can fire a nuclear missile, unless it's a kind of James Bond scenario. But in real life, no one person can fire a nuclear missile. On a nuclear submarine, you have two keys, two different people. You might have two different codes. You need two signals. And the idea is that's because one person can go crazy, but you'd have to have two people go crazy at the same time in order to fire a nuclear missile, which hopefully will protect us all. Your immune system is doing exactly the same thing. It does not want a single crazy B cell to suddenly start triggering your immune system. So it keeps this two system in check in order to prevent accidental activation of immunity and all the kind of damage that it would do. So your two signals have coincided. Your two keys have gone in, and now we're off. We're firing our missiles. And so the B cell now is activated, it's primed, and it's into action. And it does two things. The first thing it does is it divides a lot. You will remember that you have all these different B cells recognizing all these different things. So the vast majority of your B cells have done nothing. They've sat quietly. So this poor little chap who does recognize polio has now got to deal with this on his own, and he can't. So what he has to do is replicate a lot to kind of get a lot of friends who recognize the same target. So he divides a lot of times. At the same time as dividing, all these cells are also starting to produce their kind of weaponry, which is, in the case of a B cell, is antibodies. Okay, and actually these antibodies are basically soluble variants of that receptor that recognize the antigen in the first place, and they're shed into the bloodstream, into the body cavity, in order to attack whatever it was you recognize. So, for example, some of these antibodies will coat your virus, preventing it from fusing with your cells. Some might cluster the virus together so it can be more easily eaten by other cells. There are lots of different functions of antibodies, but these, if you like, are your disposable weapons. And this is happening within actually a few hours of that vaccination. So if anyone had a vaccine kind of in the last week or two? No, that's a shame. It would be quite good to pick on someone and, pat them on the arm and say, does it still hurt? Okay, but if you had one in the last few days, then this was happening right now. Those B cells are amplifying themselves. They're producing antibody. If you have an infection rather than a vaccine, this process is actually quite important in dampening down the infection early on. So it's producing antibodies in the first few days, and it's hopefully preventing, for example, the virus from spreading from your lungs to other parts of your body. So this is a kind of immediate response to an antigen. And now the big question comes, how do they achieve this specificity? I told you you've got millions of T cells, millions of B cells, and they all recognize different stuff. Like, how does that actually happen? And it happens in a quite extraordinary bit of sort of molecular engineering within your bodies. In fact, the potential of your immune system is almost limitless. So in theory, if you had enough cells in your body, you could probably recognize any pathogen that exists in the world. The slight problem is that you're limited by the fact your body is only so big, you can't get enough cells into it. But in theory, the system will work. And it does that because it has a very unique way of using its DNA. So let's take the example of an antibody. So this is what a typical antibody looks like in 3 dimensions. It's a kind of Y-shaped molecule. And this top bit, the 2 bits that stick out, is the bit that recognizes your foreign virus, for example. Like all proteins in your body, it is encoded by a gene. So you have DNA, you have genes in your DNA that encode antibodies. And now there's a problem, right? Because you don't have millions and millions of genes. So how can you produce millions and millions of different antibodies? And here is a very clever trick. The trick is because the genes that encode antibodies are modular. They're composed of different lumps, if you like, different Lego bricks. And during the development of a B cell, the B cell reshuffles within that gene to produce a unique combination. So here, if you look at the top, this is a diagrammatic representation of one of those antibody genes. And you see it has all these different little blocks, pale blue, darker blue, green, whatever. The finished gene only has one of each of those ones, but it's got a lot to pick from. And that picking process is more or less random. So when your B cell starts to grow up and become the big B cell, it picks one of these V segments up here, one of the D, one of the J, and it sticks them together. Just to make life a bit more variable, it's not very clever at sticking them. It makes mistakes when it sticks them. So as well as having variation in which segments you choose, you have slight variation in how they're joined together. And as a result, the antibody you produce from two B cells that might have been born at the same moment will be different, because the chance of this one picking the same random set of segments and this one is almost zero. And so during their development, every B cell is producing a different combination. And one of the clever things and really important things of the immune system is that sometimes when they do that, of course, they create an antibody which recognizes yourself, one of your own genes, and the body has a really good way of removing those cells before they do any damage. So it removes all the ones that recognize you. What's left is a random selection of stuff that recognizes foreign things. And in fact, because you have a bunch of different V segments, D segments, J segments, and all these different combinations, in principle, that B cell has about 200 million combinations available to it as it's developing. And so within your body, you have millions of B cells, and they all recognize different things. And that is the reason why almost anything I inject you with will be recognized by a B cell. So that's already pretty impressive, actually. But there's another really neat trick to this. So the first thing that happens, I've just said, is a B cell's been activated now. All that process has happened. The one that recognizes, our particular problem, our polio virus, for example, has started to replicate, and it's started to produce antibodies. Those antibodies, by definition, recognize polio, but they might not be perfect. So if you think about it, imagine, you've got a glove, for example. We have at home, I don't know if you have the same thing, we have a kind of big drawer with all kind of winter random gloves in. And when it's cold and you're racing out the door, you grab a glove. And actually, there are some gloves that definitely don't fit me, but there are also definitely gloves in that drawer that are not mine, but they sort of fit. And that is true of antibodies. So this B cell has responded because its antibody recognizes, for instance, the polio virus. But it could recognize it kind of like this, right, partially. But it could be better. It could really tightly recognize it, but it doesn't. It recognizes it quite well. The B cell knows that, and so over the next few days, it undergoes this process of refinement when it starts to modify antibody. It does this by a really remarkable process called somatic hypermutation. What it's doing is it's saying, okay, I've got an antibody encoded by this gene that I've rearranged, and it's pretty good, but I think it might be better. And it deliberately targets mutations in that gene to try out different combinations. And so it's evolving its antibody as it's kind of growing. And obviously, often what it evolves is rubbish. It's not as good as the original, but sometimes it hits a variation that's even better, even tighter. And this process of refinement means that over the next few days and weeks, your antibodies get better. So when you've had a vaccine or you've had a disease, your first antibody response is okay, it's pretty good. But if you come back a couple of weeks later, the antibodies you're producing are even better, because all those cells that have mutated and found a better combination are now proliferating and producing those antibodies. And this process of refinement creates really an incredibly optimal and incredibly precise antibody. Because what you end up with is a B cell that recognizes with incredible specificity the thing you gave it, because it's evolved its antibody to fit perfectly to it. And this protects you from the infection. Then the last piece of the puzzle, okay, so this is all marvelous. This has resolved your immediate viral infection, great. Not much good for vaccination, because that will protect you now, but it will not protect you in the future. So the third trick the immune system has, and that is the one that underlies vaccination, is that when it's got this far and it's recognized that it's producing these great antibodies, some of these evolved B cells realize that they've got it right, and they enter a process of dormancy. They become what we call memory cells. They shut down and they go quiet, and they remember what they've seen, and they go dormant in your immune system. And that process of dormancy, these memory cells, is quite extraordinary. A typical active B cell will live for a few weeks, maybe a few months in your body. A memory B cell, we now have actually quite good data, will probably persist for essentially the rest of your life. And so I don't know if there's anyone in the audience who was around the sort of late 1950s, 1960s. But if you were, you might well have been exposed to a circulating strain of influenza of flu virus at that stage. You will have produced antibodies to it. The world has not seen that strain of virus yet, since that point, but you will still have in your body cells that can recognize that. And in fact, one of the things I find most amazing about this is that this is now out of date, but these are my kids and my grandfather, who's sadly no longer with us. But one of the things I find incredibly amazing is that the injections, the vaccinations my children had when they were, 3 or 4, have produced cells which will hopefully still be alive and in their bodies if they make it to 98 when, like he did. And so you are being protected now by the individual cells that you had kind of primed years ago, and you will be for 10, 20, 50, possibly even 100 years. I think that's quite remarkable. Also, of course, it's quite an interesting question biologically. How does a cell with a sort of 6-week lifespan suddenly turn into a cell with a 60-year lifespan at the kind of flick of a switch? If we could figure that out, we would know quite a lot of interesting things about biology generally. And that basically is what happens when you get vaccinated. So if you've learned enough, you can leave now. But there are some other interesting things about viruses. I mean, there's lots of stuff about vaccines, I think. And there are some things, I think, that are particularly challenging and some things that are particularly exciting. So the first question, having worked all this stuff out, is, okay, so that's marvelous. So how do you actually generate this process? How do you make a vaccine that kind of works? So Jenner, when he was doing his experiments, he didn't know anything about how you made a vaccine. In fact, one of the cool things is he didn't even know what caused disease, right? He was working at a time before we knew about bacteria or viruses, or what we call the germ theory of disease. In fact, in Jenner's day, we thought the disease was caused by bad air, miasma. So it's kind of remarkable that he made this connection that sort of drove vaccination when the underlying theory about what caused disease was wrong. But anyway, we now know what causes disease. We know it's bacteria, viruses, parasites, etc., etc. So the question is, how do you turn that knowledge into a vaccine that works? And broadly speaking, there are sort of 3 ways to make a vaccine, or possibly 4, right? So what you're trying to do with a vaccine is you're trying to alert the immune system to a threat that it hasn't seen yet, so that it's ready for that threat in the future. So here's a threat, right? So if I wanted to vaccinate you against a polar bear, as by analogy here, one thing I could do, I could expose you to a polar bear, right? You would know what a polar bear looked like then, definitely. That's a fully virulent pathogen in this analogy. Slight downside of exposing you to a fully virulent polar bear is that it may not end very well for you. And this is the equivalent, if you like, of giving somebody smallpox in order to protect them against smallpox. It might work. If you survive, you'll be protected, but it might well not. And so in theory, you could vaccinate people with the full pathogen. That is something we don't recommend and we don't do. So what you're looking for in a vaccine is a way to mimic that polar bear, to trigger the immune system to recognize something without exposing it to the fully virulent pathogen. And there are various ways you can do that. So the first and most obvious way is, what about if you shoot your polar bear? Now, right, if I see this, I think I could probably recognize that top one if I've seen that bottom one. The bottom one is pretty harmless. So what you could do is you could give somebody the equivalent of a dead polar bear or a killed vaccine, okay? And we do that. We do that some of the time. So, for example, this is flu virus. So if you've had a flu vaccination, you've probably received what we call a killed or inactivated vaccine. That is the full flu virus that has been inactivated by heat treatment or by UV or something. Won't cause disease, but it has all the structures associated with the virus. And so when you give it to somebody, their immune system recognizes it. Works quite well for some things, including flu, often doesn't work very well. And the reason it doesn't work very well all the time is that your immune system is kind of more clever than that. It realizes that a dead virus is not much of a threat. Just like when you go to the museum, you don't generally run away from the stuffed polar bear. And so actually to make a killed vaccine work, you often have to do lots of other things to try and stimulate an immune response, because generally we don't respond to dead pathogens well. But it's an option. Option 2 is instead of a killed vaccine, what you could do is take something that looks really like that dangerous fully virulent one, but is much wimpier. An attenuated vaccine like these ones here, aren't they cute? So you could give someone that. And again, if you see that, you'll probably recognize that more or less. This one, much less likely to kill you. A so-called attenuated, so if you do this for a vaccine, it's called an attenuated vaccine. It's a kind of wimpy strain. So what you do in that instance is you take your pathogen and you treat it. So for example, you might mutate it with all sorts of chemicals. You might grow it for a long time to make it kind of really weak. And you weaken it in a way that if you then give it to somebody, it might make them a little bit sick, but it definitely won't kill them. But it will prime the immune system in a way that is very, very potent. And so we do that. So these are so-called attenuated vaccines. So, for example, the measles vaccine, MMR jab that most of us have had, is based on attenuated viruses, weakened viruses. This is a really, really effective way of triggering immunity because your immune system sees a live virus, it gets really revved up. The virus is just like the one that you would normally encounter in the wild, so you get a very specific response. Big problem is getting this right is really, really tricky. You need to find a way to attenuate, to weaken your pathogen that makes it really completely safe, but still enables it to grow and cause a little bit of inflammation. And that's, that sweet spot is very, very tricky to get. You have to be completely sure that the pathogen can't back mutate and become fully virulent again. And there are all sorts of kind of challenges around this, not least running the safety trials before you can put it into people is a very elaborate process. And so for that reason, these live attenuated vaccines, although we have quite a lot of them, are often quite a big barrier to kind of getting into the real world. But there are two strategies. Third strategy is you can make this a whole bunch simpler, right? One way to recognize a polar bear is if you see the whole polar bear, but another way is to find something that is related to that is very specific. So, for example, you could look at a bit of polar bear fur, which might be sufficient to identify the whole polar bear from. And we do this for vaccines, so-called subunit vaccines. So, for example, this is hepatitis B, which many of you might have been immunized about, where we take a subunit vaccine. So instead of giving people the whole virus, you give them a fragment, typically one protein from that virus that is unique to that virus. The protein stimulates an immune response directed against that one protein, but when you then encounter the full virus, because the full virus has the protein within it, you still launch an effective immune response against it. Subunit vaccines like this are really, really powerful because they're much simpler to design. You find a single protein, off you go. Completely safe because there is no chance that a single protein can cause disease. Quite versatile, you can combine them. Big problems, again, is that you've got to make the body react to a single protein, which it often doesn't do. And you've got to find a protein that, when it's present on the full virus, will still generate a really strong immune response. So, for instance, you don't want a protein that's buried on the inside of the virus. You need it on the outside. And so, for example, for COVID-19, we use this spike protein that's on the surface of the viral particle because the immune system can see that. Other big problem with this, of course, is you have to be absolutely sure that the bit of the virus or the bacteria you use is not accidentally present somewhere else, because otherwise you find that your immune system is now primed and will start reacting to things that you encounter in the world that are completely harmless but look a bit like it, or possibly even to things that you produce within your own body that also look quite like it. I'm hoping this isn't actually a really polar bear fur coat in this person. But you have to design a vaccine that is completely specific to your virus and not present in your body, and that can be kind of tricky. But we do all three of these approaches. For most of the history of vaccinology, these are the only three approaches we've done, and pretty much, hopefully, all of us in the audience will, in our lifetimes, have probably had all three of these things applied. What has changed fundamentally in the last few years, though, is this entry into this field of two other approaches for vaccine development, which are mRNA and viral vector vaccines. These are technologies that were being worked on for a long time, actually for all sorts of applications, but have not yet reached the market. And then COVID-19 came along, and there was clearly a massive focus on accelerating technologies for vaccines. And it was these two approaches which sort of came to the fore as a really rapidly deployable system. And they actually work in quite similar ways. They both work on the principle that if you want to trigger an immune response, you need to make some kind of protein and put it into a body. At the minute, those vaccine types we've been talking about are all exogenous. We make the protein and we inject it into someone. But what about if you could just make your body make the protein itself? all of the cells of our body pretty much are making proteins all the time, right? So you've got cells in your eye making the blue eye protein or brown eye protein. You've got ones that produce hair color. All of these cells are producing proteins. So the principle of these two approaches is that you could, rather than produce a vaccine and inject it into someone make their own vaccine from within their body. So in your cells of your body, you make proteins in this system here. It's quite straightforward. You have DNA, right? You have genes. You can think of your DNA as a kind of recipe book with all the different proteins you make. And I don't know if you've got a sort of a sort of family heirloom recipe book at home, maybe your great-grandmother's recipe book or something. It has all the great recipes in, but you don't want to use that in the kitchen all the time because it will fall apart. So you might have copied out a recipe, right? And that is what your cells do. So they have the kind of master copy of all your genes in DNA. But when they're going to produce a protein, they make a copy of the relevant gene into messenger RNA, mRNA here, a related molecule, and they use that as the working copy to then produce the polypeptide or the protein that you want to do, like your blue eye protein. And this is happening in pretty much all the cells of your body. So what you can do if you want to make one of these vaccines is you can hijack that system. You can use a virus over here, which contains a harmless virus, which contains DNA for a particular gene in a pathogen, like a viral protein. If you give people that virus, the virus will do what viruses do. They inject their DNA into your cells, and your cell will then use that DNA that it's delivered to produce messenger RNA and then produce a protein. But because you have written in the DNA that structure of a viral protein, what your cell is now producing is a viral protein rather than a human protein. So that's the viral vector vaccine. And this is the technology that was behind the Oxford COVID vaccine, for example. So if you had the Oxford vaccine, you've had this process. The alternative approach is that you can bypass that first step and say, well, I don't need the master copy recipe book. I can just put the working copy in. I can go straight in with a messenger RNA. Okay, so this is a messenger RNA vaccine. You put a messenger RNA in that encodes that particular protein, your viral protein. You deliver it in a way that the messenger RNA gets into the cell intact, and now it still produces the protein. So both these technologies are essentially a way to trick your cell into producing a viral or a bacterial protein. It's really important to note that when you do this, you're not editing your own DNA. These are transient steps, right? They're putting DNA or RNA into your cell, but it won't last very long, a few days, and then it degrades. So you're not changing your own genome permanently. You're temporarily using the machinery to produce a particular protein. So you've now made your protein, like a viral protein. The cell puts that protein on the surface. And now, of course, the immune system responds and says, hey, this is a foreign protein. I've never seen this before. I'm going to mount an immune response. I'm going to generate antibody, blah, 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 blah. So for probably most of us in the audience have had either the, for example, the Moderna vaccine was an mRNA vaccine or the Oxford vaccine for COVID-19. We've temporarily produced this protein. We've triggered an immune response. And now we've been vaccinated against COVID-19. So we have these kind of 5 technologies, and I think it's been a massive, clearly a massive change for society as a whole. So I wanted to take just a moment also to think about this kind of plethora of vaccines we have and sort of vaccine choices. And one of the things I get asked a lot about is about individual choices, either for yourself, should I get vaccinated or not, or for your children, should I have them vaccinated or so. And, to a first approximation, the answer is always yes, 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 choose your vaccine. But there are, I get it, there are people who are concerned about vaccine deployment and particularly vaccine hesitancy. Should I get vaccinated or not? Should I have my children vaccinated? And that largely derives from this trade-off between different kind of risks associated with vaccines, if you like. So let's think of an example of that. So, for example, this is the flu vaccine. Anyone had the nasal flu vaccine like this? Remember getting something stuffed up your nose? Yep. I see all the younger people in the audience. It wasn't available when we were little. But for the younger people, you've had something stuffed up your nose, and it's given you a vaccine against the flu. Widely rolled out now, and this is quite a good example, I think, as flu is a widespread infection. The typical flu vaccination in a year when the vaccine matches the circulating flu strain, which is what we're obviously aiming for, if you've had the vaccination, your chance of getting flu is about half if you weren't vaccinated. And if you get flu, your chance of dying from it is reduced by 30%. So the overall reduction in your risk from this one vaccine is actually quite effective. So it sort of seems like a logical choice. You want to get flu vaccinated. But you may say, like all medicines, vaccines have side effects, and that is true. Most vaccines have quite common side effects that are frankly quite irritating, like that kind of soreness and maybe you feel a bit feverish or whatever. Most of us don't mind about those. The one we worry about, quite rightly, both as recipients and as immunologists, is the one that might cause really severe problems, so particularly anaphylaxis risk. So the risk that the vaccine will trigger a very, very dramatic immune response and cause anaphylactic shock, which can, of course, be fatal. I totally understand that people are worried about that, but it's really important, I think, to keep it in proportion. So for this same flu vaccine, for example, we now have, the flu vaccine is rolled out right across many, many countries, so we have really good data. The chance of anaphylaxis is about 1 in 1.3 million doses, okay? And clearly, not all people who have anaphylaxis will die, but nonetheless, it's a scary thing. But it's about 1 in 1.3 million. To put that in proportion, your chance of dying, to have a 1 in a million chance of dying in a road traffic accident, you have to drive 200 miles. Okay, so I live in Birmingham, so my round trip, I actually took the train, but had I driven my round trip from Birmingham to London and back today, back 200 miles, I will have taken a far bigger risk with my life than I will ever take by having a vaccine, okay? And most of us drive a lot more than 200 miles in our lives. So I think it's really important to keep that in proportion when we're thinking about this kind of overall global impact. So we've talked a bit about how they work. We've talked about kind of relative values. But what I want to do for the last sort of 5 or 10 minutes is think a bit about where we might be going with vaccines. And for those of you who've been to either my lectures or I suspect quite a lot of my Gresham colleagues' lectures, you'll know that quite often we're a bit gloomy at the end of our lectures, particularly if you come to my colleague Mars Allen's on climate, you go away thinking, well, that's it, we're all doomed. And a few of mine, I have to hold my hands, I've got a few of mine I kind of usually end with the fact that, bacteria are going to eradicate humanity or, lots of other kind of negative things. But this is a unique lecture in which you're going to go away feeling hopefully massively inspired, because I think this is a huge good news story. The future of vaccines, I think, is really, really rosy. And I say that for a variety of reasons. The first one is the rate of progress in vaccinology is absolutely eye-watering. So here in the UK, this is data from the UK, here is the list of vaccines that were introduced to the UK population in over 50 years, between 1950 and the end of the millennium, okay? And there are, 11 or something, I think, right? So there are 11, which is, don't get me wrong, that was an amazing set of progress, but 11 in, say, 50 years. Since the year 2000, in fact, in the 20 years, 2000 to 2019, we introduced all of these, almost twice as many in half the time. And actually that pace, of course, has picked up since then hugely because of things like COVID-19. So the rate of generating new effective vaccines is going through the roof. And particularly these newer technologies like mRNA technologies will start to open up more and more opportunities. You'll be aware, for example, of quite promising data from Africa on malaria vaccines, which has been a really long, very, very difficult goal, and lots of others. The Ebola ring vaccine looked really promising, lots and lots of things coming online. So I think there's a huge cause of optimism around what is coming. There's also a lot of sense of optimism around delivering vaccines. So there are two problems with delivering vaccines at the minute. The first is that people generally don't like needles. I get that. The second is that it's quite a practical challenge. So to stick a needle into somebody's arm, generally you should be kind of clinically qualified. I prefer not to have my, random next door neighbor sticking it. Actually, my next door neighbor's a doctor. But, a random next door neighbor sticking a needle in my arm. And often you have to have a cold chain. You have to keep the vaccine in a fridge or a freezer before you use it, which is not a problem in London, is definitely a problem in rural Ghana, for example. So that deployability has been really limiting. However, there's some really exciting progress. So we've already talked about the nasal vaccines, which are starting to come on board for some respiratory diseases in particular. But even more exciting, I think, are things that might replace injection needles. And so there's quite interesting work around deployable patches. So this is a tiny little sticky patch, like a plaster, that has these micro needles that you might be able to see in the top in it. And the idea is that you stick that on your arm. You don't feel the needles really, because they're also tiny, but they do a tiny, tiny dose injection into your arm of the vaccine, and you would be vaccinated. And I think if we can pull this off, can you imagine how great that would be? You say, oh, I'm going to go to the tropics for holiday. I need a yellow fever vaccine, and I know your pharmacist sends it to you in the post. You stick the sticker on, job done. That would be fantastic, wouldn't it? The big goal, of course, would be to do this and use an oral pill-based vaccine. And this unfortunately, is quite tricky, and it's tricky for two reasons. One is you have to make a vaccine that will survive your stomach, which is a pretty inhospitable place. So that's not great. The second is that your gut is actually very, very good at not provoking generalized immunity, and that's the reason you can eat all sorts of foreign stuff that isn't human, hopefully, don't eat humans, and doesn't provoke an immune response. So it's not a very strong way of delivering a vaccine. And the third problem is, even if you do generate immunity, you typically restrict it to the gut. So this is probably more promising for things like foodborne disease, where we might possibly be able to generate a kind of pill-based vaccine for Salmonella or Campylobacter, but we'll probably never have a pill-based vaccine for HIV, for example. The one which I think is kind of intellectually but also ethically quite challenging is the idea of a self-propagating vaccine. So we have vaccines now that are delivered by virus. I just talked about viral delivery vectors. And we have live attenuated vaccines for a long time. So these are things which are alive when you put them in. So it would be completely conceivable to engineer those live vaccines to spread themselves, right? So it would be, at least on paper, quite theoretically possible, for example, to generate a vaccine against polio that was a live spreadable vaccine. And I could give it to this lady sitting here in the front, who's now thinking, why don't I sit in the front? And over the course of a few days, she would generate an antibody response. She would also start shedding the virus. And all of the people, I know you all came together, so all the people who are on the same journey back would get infected with that virus and become immunized themselves. That is quite an attractive idea from a public health dimension, because instead of inoculating all, whatever it is, 80 of you in the hall, I'd only need to inoculate maybe 2 or 3, and you'd spread it to everybody else. But it raises massive ethical questions, right? And I think realistically, no one is proposing this for humans, not least because of the challenge of informed consent. I cannot consent to someone sneezing on me and giving me a vaccine. And so there's a huge risk there, not to mention the challenges around what happens if it goes wrong and you've released it to the world and so on and so forth. So I think for humans, very unlikely we're going to ever go down this route. But it may be quite interesting for animal vaccines in 2 areas, I think, particularly in agriculture. So you could imagine a vaccine for Salmonella in chickens, where instead of having to inoculate every single chicken, you might just give it to 10 or 20 chickens and they'll spread it through the rest of the flock. You could also imagine it as a conservation tool. So, for example, a transmissible vaccine for rabies that you could deploy in wild dogs or wolves. They would infect each other, vaccinate each other, and you wouldn't need to make sure you kind of capture and vaccinate individual animals. So I do think there is a place for these kind of vaccines, just not in us. So that's one possibility. One of the things I think that's also coming is this idea of personalized vaccines. The mRNA and viral vector vaccines are driven by genetic technology, right? You produce DNA or RNA that matches your virus. Our ability to do that at scale and at speed is now enormous. You can produce big pieces of DNA within minutes. And so it is actually quite possible that very soon we'll start to have vaccines that are not just a generic vaccine, but are very specific. So you might generate a flu vaccine that is perfectly matched to the particular variant of flu that is in your town at that moment. You might produce a vaccine that's kind of optimized for people with immunocompromising disorders or young people or old people on a much more bespoke level than we do currently. And I think that is quite an attractive prospect. And then probably the really big one that is opening up in front of us is this idea of applying vaccines not just to infectious disease, but to other diseases too, and particularly ones that are non-transmissible, but are a huge cause of morbidity. And the really big one, of course, which is quite exciting, is cancer. And you might have heard about cancer vaccines. This is actually really quite challenging to achieve. And to be honest, I think there were many people who thought we would never do this, but the data happily is demonstrating that you can. The reason it's a real problem is that cancer, by definition, is caused when your own cells have a mutation and they start to grow out of control. But the foe, the enemy, is your own cell. So your immune system does not recognize it, quite rightly, because it's your own cell. And so the question is, how can you generate an immune response against your own tissue that doesn't destroy healthy tissue and just destroys the cancer? And the trick there is to try and find the thing that has mutated. So when a cell mutates, and this is, for example, a healthy skin biopsy, and this is a skin cancer, and you can see the difference. These cells have started to grow out of all control. That has been driven by a mutation in those cells. That mutation, by definition, will have created changes to proteins, for example, the proteins that drive cell proliferation. And so those proteins in these cells will be slightly different to those. So if you can get an immune response that distinguishes between healthy protein and unhealthy protein, you can target your immune system to the cancer tissue and deal with it. That's theoretically possible. It is now proving to be practically possible. Not for all cancers, and it probably never will be, but for some cancers in some people, what you can do is you can DNA sequence their tumor, you can identify the mutations and changes the protein, you can create a vaccine against that, and then you could immunize that person and trigger their immune response, perhaps not to completely eradicate the cancer, but in conjunction with chemotherapy or radiotherapy to shrink it and maybe even remove it. And I'm actually really optimistic about the future for this in cancer, and possibly even in other diseases like autoimmunity or a whole variety of other non-transmissible infections. So I think the future is really bright and exciting for vaccines. And so I would really encourage you, if you agree with me or even if you disagree with me, to take a look at some of the additional resources here. Thank you so much for coming. I'm very happy to have any questions.